Thank you. Sorry for the delay. I'm Yi-Seon from Professor Seung-Hee Lee's lab. And first of all, it's an honor to be here, and thank you to the organizer for giving opportunity to present my work. Today, I want to introduce about the sex-specific impact of early social experience on the maturation of Parvalbumin inhibitory circuit. As you all know, the COVID-19 pandemic forced many children to stay indoors and isolated over the past few years. This prolonged social stress affects various systems related to mental health. The experience during early life is critical not only for the shaping proper behavior development, but also for the maturation of the cortex. One of the hallmarks of postnatal brain maturation is the development of parvalbumin positive interneurons. For instance, a lack of visual experience during a critical period has been shown to impair the development of PV positive neuron in the visual cortex. So here, we aim to understand how adolescent social experience shape this maturation process and finally influence to the adult behavior. So we adopted the mouse model of post-weaning social isolation. We performed the chronic isolation from the weaning period to the adult period. The recent study suggested the sex-specific effect of social isolation, but it is not well known how females respond to this chronic isolation stress. Therefore, in this study, we targeted the orbitofrontal cortex, which is one of the prefrontal cortex and known as the social brain, and investigate how this isolation affects adult social behavior, PV maturation, and cortical network in a sex-specific manner. First, to identify the behavior impact of long-term isolation, the sociability test was conducted. So we compared the male and female mice with group housing, one-day isolation, and post-weaning social isolation group. As a result, the sniffing index and the social interaction index was significantly increased in the female post-weaning isolation group. And this did not appear in the one-day isolation and male mice. In addition, when we checked the estrous cycle of female mice after the test, there was no cycle difference after isolation. So here we conclude that this post-weaning social isolation induces an abnormal increase in sociability only in the female mice. Next, we examined whether this isolation affected the maturation of the PV positive neurons in the OFC. So we performed the immunostaining for NUN and PB and automatically counted the number of neurons. Interestingly, the number of PB positive neurons in the left orbitofrontal cortex was significantly decreased in the isolated female. So our data indicate that chronic isolation impairs PB expression in the left OFC, especially in the female mice. Then we next wondered whether the neural response to social context has been altered. So we performed the in vivo multi-channel recording. We first recorded the spiking activity in the OFC and continuously recorded by presenting a noble mice in front of the recorded mouse. During the recording, we monitored by mouse sniffing behavior, so we could track the sniffing-induced neural response. Interestingly, in group housing female mice, the neural response were stronger during social sniffing than object sniffing, especially in the left OFC. However, these social responses were largely decreased after isolation. In addition, the number of social responsive neurons and the amplitude activity were also decreased after social isolation. So this data indicate this isolation may decrease overall social representation in the left OFC. So from now, we identify sex-specific phenotypes after isolation in female. We observed enhanced sociability, impaired maturation of PB, and decreased social representation in the left OFC. But why and where does this sex difference arise? Because this long isolation period includes both the early and late adolescence with the onset of puberty, we thought this may lead to sexual dimorphic changes. For the next question, we asked how mouse sociability and PB maturation progressed after the onset of puberty in both male and females. Here we track the sociability and PB maturation changes during this late adolescence from postnatal day 50 to 80. Interestingly, we found a significant decrease in sociability at P80 in female mice, but not in the male mice. And during this period, the PB positive neuron increased significantly in the left FC. So this data suggests that late adolescence is critical window for maturation of sociability with an increase in PB positive neuron in the left FC in female mice. And we also checked the effect of direct isolation during this period. And in addition, we regrouped a group of isolated female mice at P50 to make them re-experience social interaction during this late adolescence. As a result, the P50 isolated female mice showed an increase in sociability, while regroup housing mice nor male mice did not show any changes. In addition, the number of P positive neurons in regroup housing female mice returned to the control level. Collectively, we suggested that the social experience during late adolescence was sufficient to mature both PB positive neuron and sociability. Based on the findings so far, we have confirmed that social isolation during late adolescence is crucial for female hypersociability and the PB maturation. So then, what could be the causal relationship between these two? By using CRISPR-Cas9 system, we selectively knock down the PB gene in female mice. Here we constructed the AAV virus for that and injected into the left FC at around P50. As a result, knocking down PB induces hypersociability, which is very similar to the isolation effect. In addition, when we perform in vivo recording, the PB knockdown decreased social snipping-induced activity in the left FC of female mice, which is also similar to the isolation effect. And here we found that the average baseline firing rate were significantly higher in the PB knockdown group compared to the control group. So with this increase in the baseline activity, we next checked whether this has altered the network activity of OFC, such as EI balance. Here we performed the patch clamp recording of PB positive neurons and excitatory neurons in the left OFC. In PB knockdown group, the PB positive neuron showed reduced intrinsic excitability and no significant changes in spontaneous EPSC. But when we recorded the excitatory neurons in the PB knockdown mice, it showed a significant decrease in both amplitude and frequencies of spontaneous IPSC. So this data indicate that the decreased expression of PB reduced the excitability of PB positive neuron and in turn reduced inhibition of the left OFC of female mice. For the final step, we thus examined whether this abnormal increase of the OFC activity or the direct inactivation of PB positive neuron can alter the behavior itself. To do that, we conducted the optogenetic modulation in OFC by inserting a wireless optogenetic device during the social behavior. When we optogenetically activated left OFC, the channelrhodopsin expressed mice showed higher level of social snipping behavior than the control mice. Or, when we optogenetically inactivate the PB positive neurons, the R2 expressed mice showed a moderate increasement of social snipping behavior too. Collectively, our data demonstrate that hyperexcitation of the left OFC by reducing inhibition cause hypersociability in female mice. And here is the conclusion. In this study, we found that social experience during late adolescence is critical for shaping sociability in the female mice. The post-weaning social isolation enhanced sociability while impairing the PB maturation of the PB positive neuron and decreasing social snipping-induced activity in the left OFC. And the knockdown of PB in the left OFC induced the same phenotype as the isolated mice. and these effects were due to the decreased PB excitability, which caused hyperexcitation in the left OFC. And we further found that optogenetic modulation of left OFC can induce enhanced sociability in female mice. In conclusion, our data indicate that adolescent social experience is crucial for EI balancing via the maturation of the P-positive inhibitory circuit in the left OFC to shape sociability in female mice. I'd like to appreciate Professor Seung-Lee, who advised this research, Dawn Jung, co-researcher, and all lab members, collaborators for the helps and discussions. Thank you for your attention.